Which way a cell divides, which way a cell migrates, and which way a cell ends up facing, all of these require cell polarization. A protein called Cdc42 is a known regulator of cell polarization in a wide range of biological contexts and in a wide range of organisms, from yeast to man. Alan Hall and his team at the Memorial Sloan Kettering Cancer Center in New York wanted to know if Cdc42 controlled cell polarization in the gut.
In this particular series of experiments, we were interested in how it controls epithelial polarity. There have been quite a lot of experiments done on epithelial polarity in various assays, and in some of them, Cdc42 had been reported to disrupt the ability of epithelial cells to polarize. Very unclear how that worked, and some ideas, but exactly how it worked wasn't clear.
Cells that line the intestine are polarized in such a way that their apical surfaces face the gut cavity or lumen, and their opposite surfaces adhere to the basal lamina. This orientation is critical to the cell's functions, but how is the orientation established, and is Cdc42 involved? To answer these questions, Hall's team grew intestine-like structures in culture using a human intestinal cell line called CACO2.
Most of the experiments had been done in tissue culture using epithelial cells grown on a plastic cover slip, essentially. But we wanted to try and make this assay a little more sophisticated by growing this epithelial cells in a 3D matrix.
When grown in a gel-like substance called matrigel, the CACO2 cells divide and grow into a sphere with a single layer of cells surrounding a hollow centre, much like the cavity of the gut. Here you can see the intestine-like spheres, the team called them cysts, growing in the culture dish over the course of about 24 hours. Much like the cells of the real intestine, the cells of the cyst accumulate an apical protein, shown here in red, on their cavity-facing surfaces, and a basal membrane protein, green, on their basal surface, or here on the outer surface of the cyst. The big question for Hall and his team was, does Cdc42, the known polarity-promoting factor, control the apical basal cell orientation in the CACO2 cell cysts? To find out, the team removed Cdc42 and watched what happened.
Now, when we removed Cdc42, we expected this to be just disrupted, i.e. loss of polarity. But in fact, we ended up with a 3D structure that contained not one central lumen space in the middle, but several. But each of these little lumen were polarized in themselves, but clearly the overall structure was not normal.
Proteins that typically locate to the apical membrane, such as the one shown here in green, were still found at the cavity-facing side of the cells, however. Cdc42's role was therefore not immediately clear. The team decided to look at earlier stages of cyst development. Believe it or not, polarity in the cells of the cysts starts at the very first division. In a single CACO2 cell, an apical protein, shown here in green, is located uniformly around the cell membrane, but after division the protein localizes between the two daughter cells. And the protein remains there at the interior of the structure through subsequent divisions. This is not the case, however, in CACO2 cells that lack Cdc42. Here we see the apical protein is not appropriately partitioned, and thus neither are the apical membranes. As these cells undergo further divisions, the cavities form around the mislocalized apical membranes, and thus the cyst ends up with multiple cavities. So how does Cdc42 control the correct positioning of the apical membrane? Since the apical membrane, shown this time in red, appears at the first cell division, perhaps division itself has something to do with it. The orientation of the division machinery, or the spindles, is what determines the division plane, so Hall and his colleagues decided to look at spindles in growing cysts.
So in order to get cell division to occur radially, the mitotic spindle in mitosis has to be organized essentially at 90 degrees to that radial line. And what we found Cdc42 disruption does is it disrupts the organization of the mitotic spindle. So essentially that angle now is not 90 degrees, but it's essentially random.
In these cells then, Cdc42 does not control polarity per se, but rather controls the orientation of the mitotic spindle. Exactly how it does this is the next question for the team. The paper by Hall and his colleagues is available in the November 17th issue of the Journal of Cell Biology.